Everyone, today we're going to cover the question, describe how the body detects and responds to water deficits. So my approach to this question would be to outline a detection, integrator, response mechanism. And all of these sorts of mechanisms, or most of them, are usually negative feedback loops, meaning that once the problem is corrected, it will then negatively or decrease the response so that you don't have over-response and overcorrection. So water in the body is about 60% of body weight. So in our perfect 70 kilogram male that we normally talk about in physiology, this would be about 42 litres. 2/3 of that is intracellular fluid, so about 28 litres, and 1/3 of that is extracellular fluid, about 14 litres. And then off that extracellular fluid, about 1/4 is plasma and 3/4 is interstitial fluid. So loss of water occurs from the extracellular fluid, and water can cross membranes quite easily, but solutes can't. They take a bit longer to equilibrate. And what this means is that when you lose water from the extracellular fluid, water will move out of the cells into the extracellular fluid, into vascular compartments, lost from there. Wherever it's lost from, it doesn't really matter, but it is lost. And this will increase the concentration of those solutes in the cells and in the extracellular fluid, meaning that your osmolarity will increase. Blood volume can also be associated with increased osmolarity, but decreased blood volume, such as in hemorrhage, doesn't necessarily decrease your osmolarity straight away because you're losing whole blood. So essentially what I'm saying is that your body can't actually measure blood volume. It measures tonicity and pressure as a surrogate for total blood volume. Now, the two ways that your body measures this, and I've sort of already mentioned it, is through osmoreceptors, which sense tonicity, and they're located in the anterior hypothalamus in the circumventricular organs, specifically the subfornical organ and the vasculosum organum laminae terminalis, also known as VOLT. And they respond to a tonicity change of about 1 to 2% from normal, and normal is 280 to 295 milliosmoles per litre. You then have low pressure baroreceptors, which are in the atria and great veins and the ventricles, specifically the veins within the chest, and they sense a decreased central venous pressure, which decreases inhibitory afference to the hypothalamus, meaning that you then get ADH released to try and increase blood volume. You have high pressure baroreceptors, and these are in the aortic and carotid sinuses, and they sense your mean arterial pressure. So when you have decreased stretch, you have decreased inhibition of the afferents that then also integrated into the hypothalamus, which is also then means that you have some release of ADH from that as well. Now, there are intrarenal mechanisms, but I'm not going to specifically talk about them today, but just know that when there is decreased stretch of afferent and efferent and mesangial cells, you will get activation of the RAS, so the renin-angiotensin-aldosterone system, which is a more long-term mechanism to then increase blood volume. While I'm on the topic of blood volume, so your body will respond to about a 10% to 15% drop in blood volume. And don't mistake this for 10% of your circulating intravascular volume, because really if you have a 500 deficit in intravascular space that you're not able to replete with extracellular fluid, that means you've probably lost about 4 litres of total body water, because your total body water is about 42 litres. Water leaves cells pretty easily and will diffuse out quite quickly. The reason why I say this is because when you think about blood loss from your intravascular space, that also represents blood loss from your extracellular fluid, which represents blood loss also from or volume loss from your intracellular fluid as well. Because as you lose blood from your intravascular space, you will draw blood from your extracellular fluid into the intravascular space to replete that, and this will happen over minutes to hours, and is a more acute mechanism for increasing volume. So let's first talk about osmoreceptors. So when you have decreased free water, your solutes will be more concentrated, so you've got increased osmolarity. This increases the tonicity of the blood. As the blood flows through the hypothalamus, it will pass these cells called osmoreceptors, and they have aquaphor pore channels that are on the cell membrane, and these allow water to easily enter and exit the cell. So now you've got a situation where there's increased tonicity in the extracellular fluid in the blood volume or in the intravascular space that's passing these cells. That will draw water out of the cells, which will crenate the cells. there are stretch inactivated cation channels. And what this means is that when they're stretched, they're inactivated and don't let cations in. So when there's low tonicity, lots of fluid in the cell, they're not going to be depolarizing. But then the opposite happens when there's fluid leaving the cell and they crenate, they now depolarize and let cations into the cell. This then activates sodium or voltage-gated sodium channels, which will then cause the release of antidiuretic hormone, which is a hormone made in the anterior hypothalamus that then goes down to the posterior pituitary through nerve endings and then released into the systemic circulation. So antidiuretic hormone is also known as vasopressin, and I'll talk about that in a minute. But the antidiuretic part of the hormone, it will go to V2 receptors in the kidney, which will then lead to more aquaporin 2 channels being inserted into distal collecting ducts and collecting ducts. This makes the collecting ducts more permeable to water. And there's already a medullary concentration gradient within the kidney, meaning that water will then draw out of this filtrate and urine into the medulla, be absorbed back into systemic circulation. It also increases urea transport into the medulla. The water resorption from the collecting duct also increases the concentration of urea in the tubule. The high urea concentration causes urea to diffuse into the interstitium, and this increases interstitial osmolarity, contributing to the countercurrent multiplication system. This is important because urea in general contributes about 50% of the concentration gradient of the inner medulla. So the overall effect of this is that you have increased water absorption in the collecting ducts, concentration of urine and smaller volume of urine, which will then lead to increased blood volume, decreased tonicity, and as a negative feedback, reduce the amount of ADH being secreted. Now, in high doses, ADH is also a vasoconstrictor, so also known as vasopressin, and this will help to increase your blood pressure in times of shock and hemorrhage. As another effect, ADH also stimulates thirst. So the thirst centers within the hypothalamus will be stimulated, meaning that you will seek water to then also increase your water intake and increase your intravascular volume and extracellular volume to decrease tonicity. Now, volume is indirectly measured by the body through pressure and stretch. So high pressure baroreceptors that sit within the aortic and carotid sinuses will be stimulated by increasing or decreasing stretch. So when the aortic and carotid sinuses are stretched, when there's high blood volume or high blood pressure, this will lead to increased firing up those nerve endings, so vagus nerve from aortic and glossopharyngeal nerve from carotid sinus, which will then decrease the sympathetic nervous system response and increase the parasympathetic nervous response. Where the opposite is true when there's low blood volume and you can have then low blood pressure and low stretch, there will be decreased firing, which will then lead to increased sympathetic nervous system and decreased parasympathetic nervous system. Now, what this does is it's another trigger for the hypothalamus to release antidiuretic hormone, which, as we mentioned before, ties into the previous mechanism to increase blood volume through absorption of free water. For our low pressure baroreceptors, they sit within the atria and ventricles and the great veins, specifically within the chest, and they detect around a 10% drop in blood volume. And as we mentioned, 10% drop is about 4 liters of total body water. And what this will do is, from the atria and ventricles, you will have, when there's decreased stretch, there will not be release of an atrial natriuretic peptide or brain-derived natriuretic peptide, which comes from the ventricles, not the brain. And this means that you will not get a natriuresis, so just no effect really. But when there's increased stretch, it means that you'll get a natriuresis because they're being secreted, so you'll lose sodium through the urine. They also, through other nerve endings, are integrated into the hypothalamus through the spinal cord. And what they'll do is, when there's increased stretch, they will then inhibit the hypothalamus from releasing antidiuretic hormone. But when there's decreased stretch, that inhibition is removed and the hypothalamus will release more antidiuretic hormone to increase our blood volume and decrease our tonicity. Intrarenal, I just briefly touch on. Essentially, afferent, efferent, mesangial cells and macula densa have stretch receptors as well, when there's decreased renal perfusion or decreased stretch, there'll be release or activation of the renin-angiotensin-aldosterone system, which is also going to increase your blood volume, but this will happen over days. Now, I mentioned before, in hemorrhage, you will still get activation of the bioreflex and antidiuretic hormone release, but your osmolarity will remain the same. So the question arises, which is more important? Will your body protect volume over osmolarity or osmolarity over volume first? And the answer is that your body will protect volume before osmolarity. So if you had completely normal osmolarity and you're having just this one-to-one loss of fluids from the body with solutes, your antidiuretic hormone response will still be activated and be quite strong with that volume loss and will override osmolarity derangements. So even if you were hyperosmolar, it would still override that. Just as a side, the order that your body protects itself is volume first, osmolarity, and then pH. Thanks for watching. If you have any questions, anything you want to add, anything you want me to cover, please let me know in the comments.